I think we should go ahead and get started. So thank you all again for joining us today. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have representations from a data coordinating center or DCC or any of the other programs that are a part of the CFDE and the centers that support them. So thank you all so much for joining. Before we get started, I have a couple events and updates from the CFDE, and then we'll get into our featured presenters. So first, I just want to remind everyone that if you're unable to attend the previous webinars, that we have them all available up on our YouTube. You can access them directly from the QR code on the left here or through this URL. And again, we'll have it underneath this CFDE specific playlist. So all of our videos are recorded there. This will be shared after as well if you would like to forward with any interested colleagues. And as a reminder, if you have any feedback, questions, or would like to be added to our listserv, you can email us at help@cfdeknowledge.org, and that'll come directly to me. Here is our remaining 2026 webinar schedule. Today we'll have CM4AI, and you can see the upcoming webinars for the next few months. We still have an open availability for November 20th, so if you're interested, please feel free to contact me. We are extremely excited about this lineup. We invite you to join us on June 29th, Monday, for a virtual roundtable with the NIH's All of Us research program. Studies here for diabetes at scale using genomic HER survey and wearable data for over 600,000 All of Us participants. And so this event on Monday is from 1 to 2 p.m. Eastern. And in this session, you can hear from researchers that are using All of Us data. And so two of our colleagues here at the Broad, you can hear from Satoshi and Julie. And at this session, you can learn all about the All of Us data and how you can use it to study diabetes at scale. This can show you how to explore real-world evidence, genomics, and longitudinal data. And we're happy to have a perspective from all of the participants on how you would use this for your own research. So again, if you're interested in joining us, you can scan the QR code on the left here to register for this event, or you can use this code at the bottom in order to RSVP. So we hope to see you all there. Just this past month, we had the Festival of Genomics in Boston, and the CFDE participated in both a booth and ancillary session. We just wanted to say thank you to everyone who came and showed up at the Festival of Genomics, but also to thank our incredible speakers and presenters at the booth for making this event so successful. We're really looking forward to the next conferences that we have upcoming. And our next one is the Intelligent Systems for Molecular Biology, which will be in Washington, D.C., July 12th through 16th. And here you can see some of the organizations that are going to be available at this conference to interact with. There'll be a combination of short talks, posters, and different presentations during certain tracks at this event. I'd also like to highlight that Julie Juergens from the Knowledge Center will be presenting a short talk, The Common Fund Data Ecosystem, Incubator for Pan-NIH Resources, Enhancing Interoperability. And this talk will be during the BOSC track on July 15th from 2:40 to 3 p.m. And she'll also have a poster session throughout the week as well. So again, if you're going to be joining at ISMB, please keep an eye out for all of these incredible organizations that will be available to connect with at that event. As you know, in 2 weeks, the CFDE is hosting the Scientific Interoperability Incubator, and this will be on July 9th and 10th. And so to prepare for this exciting session that we'll be hosting, we'd like to get as many people in our scientific orbit to share perspectives on what scientific interoperability means to you. So I invite you to fill out this short survey, and you can scan the QR code here to participate. It should take about 3 to 5 minutes to complete. And this will be open until next Monday, June 29th. And please feel free to share this survey with any of your colleagues as well. The results from this survey specifically are going to help guide our discussions and sessions at this event. So we're really looking forward to hearing from the community. And if you have any questions about this or access, please let me know. It should be available to all who scan the QR code. Um, if you are curious to learn a little bit more about the CFDE, we invite you to listen to the CFDE Training Center's Decoding the Data Ecosystem podcast, which is hosted by Dr. Alyssa Dillman from the Training Center. This podcast is more than just a podcast, it's a community for those that are passionate about the potential of omics research and using it to solve complex biological puzzles and addressing pressing health challenges. So this is for those who, could be a seasoned researcher or a student just starting out. It's for anyone who's eager to learn more about valuable insights, inspiring stories, and advice to your journey through the world of omics. So if you're interested, there are 18 episodes out now, and they are all available on anywhere that you get your podcasts. So with that being said, I just want to say thank you again for everyone for joining us. I'm going to hand it over to our featured speakers who are from the Cell Maps for AI or CM4AI. And so with that, I'll stop sharing and hand it over to the team.
Right, great. Thank you so much, Mackenzie. I'm going to just bring up the slides, and we have 4 speakers today that are going to talk about various aspects of the CM4AI project today. So we'll have Jake Chen first, providing a brief overview, and then Dr. Tim Clark speaking about a little bit about the standards and interoperability and AI readiness. And then we have Jan, who's going to be talking about imaging, and then Wade, who will kind of wrap us up with tools and training, learning aspects and training opportunities upcoming that deals with our data. So let me just bring up the slides.
Great. And I'd also like to remind everyone that you can use the chat or Q&A to ask questions throughout this session. We are going to have these questions addressed after each speaker since there is a few speakers. So we'll be mindful of the time, but please submit any questions that you may have to our speakers.
All right. Well, let's get started. So my name is Jake Chen. I'm one of the PIs for the CM4AI project. Today, we are really honored to present our work to the CFDE community. I think it's special that we have investigators and scientists together to present you this a new resource. This resource has been planned since 2022. But if you also include the planning of the teaming aspect, we actually go back to the end of 2020. And so it's participated by many institutions. We listed in the cover page. Primarily, this is a data generation project of the bigger consortium called the Bridge to AI program. And the goal of the NIH Bridge to AI program is to set the foundations by creating well-curated data sets that are going to become pillars for the future AI growth. As we can see, today's AI is growing so rapidly, and the moat of AI future growth is actually in the data. So I think what we are presenting here today is what we call the cell maps for AI. And why are we presenting cell map? Cells are the basic components. If you want to think about the diseases, it's very important that if you go to the first principle is to understanding how the genetic basis of a disease, which has been mentioned by many of the genome sequencing projects, but also the dynamics inside the cells. How are proteins being expressed? How are they distributed? And if we use a different instruments, maybe we see different aspects. So the whole purpose of the cell maps for AI is to create this a foundation of multimodal understanding of what's going on in the cells, so that all of the community can benefit and then start creating AI applications. So I think I'm going to provide an overview, and then my colleagues will provide more details. Next slide.
Yeah, I'm sorry. I'm trying to advance it, but it's not moving. One
Okay.
second.
Um,
One
as
second.
you're, yeah, as you're waiting, I think you can see that we have multiple talent. So Tim, who will be joining me, is from University of Virginia. He's an expert on ontology curation. Yan is a scientist from Stanford University, is involved in spatial uh, high content cell imaging project, and we will talk about an exciting new modality and some of the early work. And Wade from Yale University is going to talk about all of the training aspect. How do we take this data set and encourage the community to think through various applications? Are we able to advance now? Or
It's not
okay.
working for some reason. It was working earlier. Let me try
Yeah.
one more time.
I was gonna say maybe if you if you exit the screen share and then maybe can somehow exit full screen of the PowerPoint and then maybe restart it somehow.
Yeah, I'm gonna close out of the PowerPoint completely and try again.
Well,
Let's see.
as you're preparing, I'll just keep on talking. I
Yeah, I'm
think
going.
this project we're participated by multiple institutions, and our data come in multiple modalities, right? So to give you an idea, in order to understand what's going on inside the cells, uh, traditionally people only measure the RNA sequencing, so measure the RNA transcript. But in this project, we decided to actually go in a lot more details to measure what's going on inside the cells at the protein level. Because we believe that the RNAs, and that you mostly just destroy the cell, measure the RNA content, and then you lose the resolution of how the cell, the transcript are spatially distributed. So we would like to preserve that. So as you can see that the overall uh, cell map for AI is to go from beyond the genome sequence to look at the molecular structure and function and their distribution and their perturbation inside the cells and tissues in order to understand cellular conditions, disease, and the response to drug. So particularly for us, we look at the two cell models. One is triple negative breast cancer cells from minority donor. And we subjected to two drug perturbation, chemo drug perturbation conditions, and measure all of the modality of the data. And another cell lines that we use is a human induced pluripotent stem cells, iPSCs. And we try to differentiate them into neural cells and the cardiac cells, and also measure the different aspects. In terms of the modality, we measure these three broad modalities. One is that as the technology advances, we can look at the use of confocal microscopy, look at the multiplex protein distribution inside the cells. So essentially looking at the subcellular protein distribution in the multiplex fashion. And then we use that data, we capture that data. And Yang will talk about that in more details. Another modality is to look at the protein mass spectrometry data. We use systematically curated the bait proteins that are important for chromatin uh, reorganizations, and these are considered to be systematic targets. And then we use those as a bait and to look at what kind of different preys that can be fished out in a very complex system and then read out that data. We also use a single cell RNA sequencing, of course, but then we also, this is done by UCSD, Prashant's group. And then what we are trying to do is to look at all of the RNA sequencing data under different drug perturbation or just CRISPR gene knockout conditions. Next slide. So a little bit of a rationale why the multimodality is important. This is one example that was published in the UCSD Trace group. So if you look at the collagen, for example, collagen is highly structured fibers, and that has lots of collagen fiber structures, and then the fibril and the microfibril structures. If you look at just the collagen protein or RNA distribution, you do not understand fully what's going on. And you can probably just get the protein network. Can you just move on with the animation? Yeah. But actually, if you also tend to have the imaging data, right, you can see that on the upper panel, the imaging of a normal collagen complexes versus the imaging of a mutated collagen matrix, you see dramatic differences in the distribution of proteins in different cell types. So adding this modality allow you as a scientist to infer what may be going on in, for example, cancer context, the potential changes, and give you a lot more context than protein interaction information alone. Next slide. And you can also take another example of this structure called the decimal sum. And the decimal sum is like intercellular structures that is very important, and that's involving multiple proteins. And in fact, if you try to understand such a complex scenarios, the only way to understand it is to look at the spatial distribution at the protein level, right? And then this is one example that you may potentially understanding disease biology through multimodal subcellular information created from cell models. Next slide. I think that CM4AI, with the past 4 years and multi-group effort, we have created some what we call the flagship data sets. It's a flagship data set because if go to many of the data repository, it is fairly easy to find a data set that is a single modality, right? But it's fairly rare for you to find the multiple modality all controlled under the well-coordinated conditions. For example, here that we have this human iPSC cells. Today, if you go to the data repository and the CM4AI.org data releases, you will see more than 11,000 genes under perturb-C conditions. And then you can see more than 7,000 proteins are captured in the mass spectrometry data, and lots of the confocal imaging information, both the raw and I think the process information. I won't go through the details, but I think that this is the data that I listed here is still being updated. By the end of this year, we will have the most complete data available completely for free for the community to analyze and think about. And if you actually last through the end, and the Wade from our group will tell you some exciting challenges that we're putting forth. I won't be spoiling, have a spoiler. Okay, yeah, next slide. So this is another recap of the type of the data that you can look at. And if you look at all the raw data, I think it's a tremendous information. Lots of them are imaging information. Next slides. I think this project, we have several modules. There's an exemplar by NIH, how the future AI or large collaborative project at the national level should be done. It was not an easy ride, but then I think we're very proud to say that this is not just about data generation. We also have a module that's called a tool modules, where people from Trey Adeker's group at the UCSD are creating really exciting, innovative visualization. What is shown here is visual AI that Trey would put it, is using gene ontology in a hierarchical way and create visualization so that you can know in a glimpse what gene ontology categories are being enriched and different from one data set to another. And in fact, there will be visualization context, and we're continuing to look for additional visualizations. That's wearing my hat as a CFDE Integration and Coordination Center. You're welcome to take a look at it and then say, well, I have better method to look at this data set as well. Okay, next. And I think that we are not really doing all of the AI and machine learning, but we are in the process of creating models for the community and to demonstrate that we as an academic group can create foundation model, can benefit the broader community. As many of you know that even last year's survey, I found out that there are more than 200 foundation models in life sciences. Why do you need to create a new foundation models based on CM4AI dataset? But you probably will be disappointed if you look at out of the 200 models, many of them are commercial, and they're probably being fed with a data set that we don't have access. And it's very difficult to evaluate the performance and contribution to science. So I think this model, Yan will talk about in detail in his talk, is one of the effort that we're trying to make it openly accessible, that make use of the imaging information. And the people, I believe that the PI is Professor Emma Lemberg from Stanford. And I believe that they're going to make it available to all of you. All right, next. So I think imagine that if you have a data set, and we're already brainstorming what you can do with the data set, right? So this is one of the Trey's example. He's trying to study breast cancer and try to set up a hypothesis of this particular protein called the SMAC-A4 protein may be implicated in breast cancer. And this is one example that you can actually look at the different modality of data and then see what's going on. You see in the protein imaging, in the normal situations, and it's not detectable. And that means that is not expressed. But somehow in cancer cells, and they come out. And that's the first thing that you see. Oh, yeah, that's a very strong correlation, much more than just expression level differences. Then when you look at the protein interactions, you will see that the SMAC-A4 is not an insignificant player in the network. And when you look at the the protein level, the proteomics data. Likewise, you get the confirmation that is a bigger node. Through those examples, and then I think even trivial examples, you can start hypothesizing, maybe even before I even integrate everything into fancy AI models, I can start doing some hypothesis-driven biology. Right, next slide. So I think we're just trying to show you that to generate this data set, it requires lots of the hidden effort, and the Tim will be showing you all of the details and behind the scenes, how we're accounting for individual data sets. And then we're keeping track of, what the community is actually needing, and then try to do a good job so that when you go to the website, and we want you to understand fully what this data really meant, and then you can actually download the data and then publish papers. So if you want to just a published paper, have great experience or bad experience, we'd like to hear. So in fact, for this project, one of the roles that I didn't mention yet is a teaming. So teaming is considered to be important, and not only to team among ourselves to make sure that we are well coordinated, but also to the community. We're going to roll out new software tools, and we're not going to talk about it here today, but if you register for our portal, over time, we're going to roll out those exciting tools that allows you to collaborate with other people, even if you're just an AI researcher. Next. Yeah, so go ahead and visit the website, CM4AI.org, for the tools, and you can visit the Cytoscape, and then that's it. Yeah.
Well, can you back it up a little there, Swati, back to
Yes.
where it starts? So just let me point out an issue that many people are concerned about, the interpretability and explainability of the results of AI models. And as we know, these deep learning models and generative models, they're very, very complex, and it's completely unclear in many ways. It's opaque how they compute their answers. So there are a number of techniques that were developed called post hoc or post model explainability to infer how the interpretations were done. But what people have left out, and what's very important for us, is looking at the pre-model transparency. That is, in biomedical informatics, the the processing pipelines are long and complex, and CM4AI is probably a great example of this. And what happens in the pipeline and decisions that are made in the software and data set selection and all the rest of it, sample selection even, have great impact on how things are computed downstream and what answers you're going to get. So, going back to a slide that Jake showed, we're taking lab samples and instruments, computations, and data sets, and taking the raw data, transforming it into interpreted data. Then we get protein-protein interaction and image embeddings. Then we go into the cell mapping toolkit, and you do more transformations. In fact, in this pipeline, we actually use AI to compute the embeddings on the protein-protein interaction networks and the image localization networks. And then we combine the embeddings and do community detection, validation, distance computation, and then the visualization. So this is a complex pipeline, and we would actually like to know what the heck is going on in there. So next slide, please. As we noted, we compute these hierarchical cell maps showing the decomposition at various levels, from the cell to the subcellular organelles, down into individual protein complexes. And if you'll notice that quite a few of these are putatively novel complexes, right? The purple ones are putatively novel, which means they have to be investigated. So we would like to be able to have some interpretability to see what are the most interesting ones, what are the ones that have the best evidence, and so forth. Next slide. We built a software toolkit or framework called Fairscape to include metadata standards at all points in the data acquisition and integration pipeline. You can see that all the labs that are involved generate data sets that are rich local metadata is attached, and the metadata and data packaged together in standardized packages. We use the RO Crate standard, Research Object Crate. It's used at Jackson Labs and in a lot of places in Europe. And they're fed into, they're integrated through the tools pipeline into one common package, and that those are uploaded into a server that produces a fair data archive. Next slide. And we developed our framework in close collaboration with the Bridge to AI Standards Working Group, which I co-chair with Professor Monica Munoz Torres at the University of Colorado. And this Standards Working Group developed AI readiness criteria for all projects in the Bridge to AI. They're applied to all the projects, and we have assessment tooling to score that, right? So the AI readiness consists of fairness, provenance, transparency, deep characterization, pre-model explainability, ethics, sustainability, and computability. And the paper out on that that is we've just revised it, and it's been submitted to scientific data. We're waiting for a decision on it now. Next slide. Fairscape establishes the AI readiness metadata for each of these criteria or axes. So we apply the fair principles or fair criteria from Wilkinson et al. We produce provenance graphs, resolvable provenance graphs. We produce deep characterization, describe how ethics were reviewed, what the HL7 security requirements are, licensing, computability, and all the rest of that. And so that's really important for people to be able to know what the potential use cases and prohibited use cases or discouraged use cases are for this data, that it's valid, and how it was developed. So you have transparency. Next slide. We do this as part of the Bridge Center Standards Groups or the Bridge AI Standards three-layer metadata architecture, so that the people working in the domains use familiar tools, familiar models, that we have a canonical exchange layer. These RO Crate packages include the W3C prob and EVI provenance graphs, Croissant and Croissant responsible AI metadata, and also a base translation into LinkML semantics. And the deep semantic layer is a work in progress. We're just finishing this shackle characterization or shackle definitions, and we're working on other things. But this whole framework is reusable and something that anybody can apply. Next slide. So we produce these metadata, and then we run evaluations on them and score them. And we don't produce, we do produce a single score at the end, but of it's composed of multiple scores, right? Next slide. And we produce human readable data sheets with each of the data sets. These are actually very extensive and treat each different data modality independently as well as in aggregate. Next slide. And at the end, you get one of the important things is these provenance graphs, which are in JSON LD, which is, as you know, an RDF serialization, and each node is resolvable. So a data set node here is resolvable to the actual data sets, primary and intermediate data sets, and the software is resolvable to the software archives, which gives you the ability, if you do the resolution, to compute audience focused interpretability. So, what would a clinician want to know? What would a biostatistician or a data scientist want to know, and so forth? So, these tools are open source license under Apache 2, and we definitely encourage people to use them and to apply the AI readiness criteria, which we have packaged and evaluated all four of the Bridge to AI program dataset productions using these standards, and we hope that that's a big contribution to the community.
Yeah, hi, it's my pleasure to be here today. My name is Jan Hansen, and I'm a postdoc in Emma Lundberg's lab. I'm going to introduce you to the spatial proteomics side of this project. And we at the Human Protein Atlas, in particular Lundberg Lab, have been running a section in the Protein Atlas that focuses on subcellular structures and where protein localizes in individual cells. And what we see here is one of the example images, a cell with a nucleus, and now we see the microtubule cytoskeleton stained. And now this will be the ER. And then we will see a protein of interest. In this case, it stains the plasma membrane. And this has been a many-year effort in the Lundberg lab, where we basically stained with robots and the antibody library from the Human Protein Atlas project, one protein at a time, with the three reference markers that we just saw: nucleus, microtubules, and endoplasmic reticulum, or ER. And so this is a tedious effort, and over years we have stained many proteins, but now we try to bring everything together in a single cell. And we have just released on bioRxiv, so you can access this preprint now, and we also share the model with you. And what we in the essence did is we trained a generative model that allows us to, just based on these reference markers that we just saw, ER, microtubules, and nuclei, paint a protein of interest. And this has been a big effort by a computer science postdoc in our lab, Paul, together with others, to make this a really applicable tool, which we now are happy to share with you. Yeah, so what we see here on the right, oh yeah, we can move on. That's good, Swati. So what we see here now is that this works not only just for a single cell like we just saw, but we see it here labeling all kinds of different cell lines. And in the end, Prodicelli, how we call this generative AI model, allows us to take a cell line or a cell image of pretty much any cell line and then paint the protein staining patterns for many, many different proteins. So we can now overlap where these proteins localize and see them in different cells. And in the essence, you can see it in a way that we transition from an atlas of proteins, where you look up a protein and you see where it stains in a cell, to taking all the knowledge that we have gained in the Human Protein Atlas and putting it into one cell of interest. And this could be, in principle, the cell that you are interested in. So next slide. So in the CM4AI project, we have been engaged in doing the spatial proteomics side, where we image a protein at a time and reference markers for the different cell lines, such as for breast cancer cells. And what we see here, for example, is MDA-MB468 breast cancer cells. We see a single cell or a few cells in a smaller crop region. And we've been wondering whether we can now use our model, Prodicelli, to predict the staining in the cell line that has never been in the training data. And so what we see here is on the right for both of these proteins, the real images that we stained. And then on the left, we try to just use Botticelli to stain the same proteins on these cells. And as you can see, this worked surprisingly well. So for the left protein, MECP2, you can see that the generated images from Botticelli, which has never seen this cell line or the drug treatments, they look very similar to the real images. But certainly there are also always some errors, which you can see on the right. So here you see that some of this looks reasonable, but you see that in the top row, paclitaxel treatment, you can see that the real image shows not a nucleoplasm staining, but the generated image imputes a nucleoplasm staining. And this is certainly also where the boundaries lie of this model, because we cannot, for now, impute something that is not in the training data. So when we look into BPTF stainings and other cell lines, which the model has learned from, there was never like a cell where there was not a nucleoplasm staining, but this more cytoplasmic membrane staining that we see in the paclitaxel treated cells. So all in all, this is still like a great advancement, because it means we now have a model where we can predict a staining experiment before we even do it, but it has boundaries, as we see in this example case. Let's go to the next slide. And other boundaries that we noticed is certainly when we use such generative models, it becomes more challenging when we look at single cell heterogeneity. What is single cell heterogeneity? So in fact, protein localization is not the same in every cell that you look at, but there are factors that affect where a protein would localize in a cell. And one really like clear example is the cell cycle. So over the cell cycle stages of a cell, proteins relocalize to different regions, or there are, for example, cell cycle regulators that we see here. So here we look at stainings of CDT1 or geminin, which are two main cell cycle regulators or regulated proteins. And on the left, you can see different cells, and they have different levels of these proteins visible in the nucleoplasm. So now when we try to use Botticelli to stain this on the new MDA cell line that we have in the CM4AI data, and this is the leftmost plot that you see, we cannot see any dynamics of CDT1 or geminin proteins across the cell cycle. So G1, G1S, and G2 phases, they all look pretty similar. So we can see that the model could not, from the limited training data, learn this dynamics. But then when we add images, so we added like I think 100 images to the training data and fine-tune Botticelli for these proteins, we can see that the model can learn to really like predict the cell cycle dynamics. So you see on the right ground truth data, on the left like what the model did without fine-tuning, and in the middle plots you can see that we now capture the cell cycle dynamics. The geminin is high in G2 cells and CDT1 is higher in G1 cells, and that these data match pretty closely to the real ground truth data. So Botticelli can also be seen as a foundation model for retraining to whatever purpose you have in your individual experiment, and then getting a really well-fine-tuned model that even can capture single cell variability. Let's move on to the next slide. Another way how we imagine Botticelli is that, for example, we look at can we now use Botticelli to disentangle gene sets? Because we all know that if we look into UniProt or ensemble gene sets, we often see that there's a lot of proteins annotated, but we would not understand their subcellular relationships. And the same holds true for many protein complexes that you can look up. So the purpose here was that now that we have Botticelli, we can, for example, take a cell type, and we see here on the left, different cells. And then we predict all the proteins that are annotated in this reactome term, metabolism of lipids. And then we go into each pixel and extract this as a vector of the different protein intensities and cluster the pixels. And what you can see here is what we end up is with different clusters that represent different regions in the cell. For example, cluster 1 is plasma membrane, cluster 3 is the cytosol, then we have a Golgi apparatus cluster or the nucleolus cluster. And now we plot the intensity of the predicted proteins. And yes, you can see here that we can use that to get a bit more spatial resolution of gene sets that we have annotated in many different databases. So let's move on to the next slide. Before I give you a summary of all the different things that Botticelli can do, because there's many more that you can read up in our paper, I want to come to one of the most important for CM4I things we did with Proticelli, and that is that we tried to build single cell multi-scale maps. So you have been introduced to that we can integrate different data to build a map of the cell. What we try to do here is that for now we use only image data, and this is certainly extendable, and I think Wade will talk a little bit more about it. But we use single cell image data that we generate with Proticelli for 12,800 proteins on a single cell. And then we try to hierarchical cluster in an embedding space all these different proteins. So we paint 12,800 proteins on a cell. We embed them with a representation model, and you're very welcome to read more about our foundation representation model in the Gupta et al. Preprint. And then we do hierarchical subclustering to let the cell fall into different organelles. And you can see that it sort of does that if you pay very close attention. We have, for example, some proteins that group into cytoplasm or nucleoplasm, and that further disentangles into mitochondria, into the microtubule cytoskeleton, and so on. And then what now we can do is if we do that for many different cells, and we did that for 200 cells, we can compare these single cell whole proteome predicted maps and see at what complexes are stable and what are dispersed. So what we mark here, for example, on the bottom is that we take a more dynamic and a more stable cluster, and now we show you how these proteins look in all these different cells predicted. And you can see that some of the proteins, they're very stable. So like we have a very stable microtubule cytoskeleton cluster on the very bottom. But in the second row from the bottom, you can see that there's more variability within between cells. So this model allows us to transition to really like build single cell multi-scale maps. And with that, I think I'm at the last slide. If you move forward. Yes, I can encourage you to look into this preprint that we just put out. We have different more applications that you can do with such a generative model. And yeah, you can even use it yourself. We have a GitHub repo where it's explained how you can apply Proticelli yourself. And yeah, give it a try, and I look forward to getting feedback on it.
Great, thank you. And the last part of this shouldn't take too long, but just wanted to walk through some of the different resources that we have, as well as some of the events that we'll be hosting throughout the rest of this year. As you've probably seen from all of the rest of the presenters, building out cell maps is fairly complex. There are a number of different raw data sets and very different modalities, and a number of different touch points where we use AI to then take those raw data and ultimately build out the cell map at the end. The resources that we have, one, you can go to our website, CM4AI.org, and then there's a training portal. We also have a YouTube channel that has introductory videos on overall what are cell maps, how we make them, and then also how they can be used. And then there are a number of code tutorials and prior challenge examples on our GitHub repo at github.com/CM4AI. Next slide. And for me, one of the exciting focuses within the cell mapping project is really where we can apply these tools in downstream applications. And so there are a number of places where you can use the raw data to build the excellent models that Jan went through, where we can build out new approaches to developing standards and apply those in many different areas. For me, being on the clinical as well as the research side, being able to take these cell maps and actually predict the impact of genetic mutations on drug response or on clinical outcomes is one of those very exciting areas, especially because of the explainability inherent in these models. The figures here are actually from one of Trey's publications, predicting drug response synergy using a deep learning model. There's actually two versions out there, one of them drug cell and a newer one, the Nest VNN. And this forms the basis of several of our tutorials and challenges where you can actually use that cell map to design a neural network architecture and then predict something like drug response based off of that mutational profile. Next slide. This just covers, again, some of those different tutorials that we have. We've got some of these on analyzing immunofluorescence imaging data, also using some of the more advanced models, as Jan mentioned on his prior slides. We also have a few of these that go more in depth into the actual systems and network biology. And so for building out how we represent the different data types, generating embeddings, generating the graphs, and places where you can tune that model across the entire pipeline. The entire CellMap pipeline is available as a set of Python libraries, and if you go to those GitHub repos, there is one full example set that takes you from end to end, and once you've developed that cell map from the raw data, can then be fed into those visible neural networks to do different predictive tasks. And next slide. A few of the prior competitions that we've done, we've hosted a few of these online using Kaggle. We've also done a few of these in person, both at Yale and at the University of Alabama in Birmingham. One of these focused on developing out improved methods for protein network community detection. So after we get all of those protein similarity scores or interaction edges, how do we actually go through and move from protein up to subcellular complexes and ultimately up to the cell? There are a number of tools that are already built into the CellMap pipeline, but there are many others that are available and could be used or substituted in an attempt to build out better networks. Another area that's been very interesting is being able to apply large language models to gene network annotation. So one challenge that we often run into is when we develop out or build out these cell maps, there's not really a gold standard. So we're taking all of the experimental data, assembling the map. We can compare it to things like the gene ontology and to the quorum ontology, but the goal here is to find new and novel associations, and there's some other example code where we can take LLMs to try to infer how these different proteins and clusters may be biologically related and give us some sense into the quality and performance of the cell maps that are generated. And then the other one, which I'll go through in a bit more detail on the next slide, is drug response prediction with visible neural networks. That was that initial use case with DrugCell. But if you move on to the next slide, we just hosted and finished another hackathon event up in Boston about a month ago now. And for that project, what the participants in our group did was take the existing Nest VNN and adapt that to be able to train not just on cell culture or sequencing data that you might have experimentally and then look at drug response in cell culture, but instead built out data downloaders to grab gene profiles from tumors in cBioportal and then be able to predict actual clinical outcomes. So did patients have a longer or shorter survival pathway, and predict that and get the explainability based off of their actual tumor genome sequences. And the nice thing with that approach is that it can actually be tuned using different input cell maps. So for predicting breast cancer, you might want to look at a cell map from a breast cancer cell line. For doing more all comers in cancer, that might be like the Nest VNN, which grabs a large number of different genes that are related or involved in different types of cancer. But from cBioportal, you can then pick a study, a tumor type. They built it out so that you could actually pick different types of endpoints, continuous or categorical. And once that was done, integrated other CFDE data sets to provide likely or potential drug targets that might be effective, as well as provide the explainability of which genes and cellular pathways were the key drivers of the individual's outcome prediction. We'll be hosting then a few other challenges, and if people are interested in these, once we finalize dates, those will be posted on the CM4AI website. In August, we'll be doing a virtual cell challenge based off of the GEARS model architecture, which you can use to predict transcriptional response to gene perturbations. The nice thing about that architecture is that you can actually embed prior knowledge about protein relationships to try to improve the predictive accuracy. And so that will be one that will be coming up in just a couple of months. In October, we'll also be hosting a quantum community detection challenge. So one project that we've been working on within my research group is, instead of using some of the more classical models like Leiden and Louvain for that community detection step, actually using quantum algorithms and actual quantum hardware to see if we can get better and higher quality cell maps and identify new and novel protein relationships. The plan is to host that event at Yale University, and we do expect to have some travel awards available. And again, that'll be the details posted on the CM4AI website. And then the last one is going to be a dream challenge hosted this fall as well. The focus of that is going to be on largely what Jan just presented. So trying to predict cellular imaging and protein localization using foundation models. That event is being hosted in collaboration with Anvil to have compute infrastructure to be able to train and implement those models using CM4AI dataset with very nice, clean holdout data sets as well for the evaluation side. And all of that one will actually be posted on the Anvil webpage and likely some announcements from the NIH as that gets finalized. I think that is it.
Yes, thank you all. So this was just one last slide that I added just for the end. I know we talked about a lot of pieces of our project and all the different components. So this QR code, if you scan it, will take you to our website where you can, revisit what's been shared here today. Just as a recap, there's a lot of tools and different datas that you can find on our website. And as Wade mentioned, you'll also be able to access all of our upcoming events there as well. So thank you all.
Yeah. So we can take some questions, and there's a question for Yan. Which version of HPA do you use to train the Proticelli model?
Yes, I was about to start reading it. Yeah, so now I'm almost like on a blank spot, but as far as I know, it's like the version 24 where we started and we were about to transition to 25. It's in our paper, it's written which version it is. I think it is version 24. And you were asking what is the public bulk export from Protein Atlas? Um, the cell line and protein meta ID metadata per image. So you can download the HPA data by creating links through the XML files for each protein. Maybe this is like a good chance where you could send me an email and I can like guide you a little more on your questions. I think that might be an easier way to communicate it. And there's also resources in the HPA data webpage itself, how you download data, but I'd be happy to guide you to find the data that you need. And then the last question is about the Kaggle challenge. Um, I'm not aware of that you can map back the Kaggle data to the protein atlas unless there's data like cell line and protein ID labels there, because this is a separate, or like to large extent, a separate evaluation set, for example, and a separately annotated data set where we go to single cells. It depends also a bit on the Kaggle challenge. But like I said, I think the easiest would be like this is very specific questions, and I'm happy to help you like find your way through, but I would also like need to understand a little more of what exactly you want to do so that I can guide you in the right way. So maybe I can just put my email in the chat and you send me an email and I'm happy to like give you like some more clear instructions.
Yeah.
And I
So
think some of that we maybe have in one of the tutorials on GitHub as well, because I know we had done some work with those older models. So happy to help out with that as well.
yeah, so there's a second question is more of a executive decision that was decided at the very beginning of the project. We understand to build a multimodal data set at such a national scale require lots of resources. And therefore, we initially proposed the 3 cell types, and then eventually we settled on iPSC due to the potential impact on the stem cell research and the neurodegeneration impact. And for cancer, we thought that if we can only do one cancer line, why not just find the most difficult one that doesn't have therapeutic solution. And we're not trying to be comprehensive and solve all the problem, but we're trying to demonstrate the power of multimodal technology, and hopefully there industry collaboration partnership that can take on from now and then do more cell types.
Great. Thank you to CM4AI for presenting and joining us today. We are at time, so if you have any other questions for our featured speakers, please feel free to email me at cfdknowledge help email, or I just put in Jan's email for any other questions. But we thank you so much. We'll see you on July 31st. So thank you, everyone.
Thank you.
Thank you.
Bye.